In lecture, we were just finishing up talking about different types or different groups of media. And if we need to, you can skip ahead of this if I got through all of this. But again, we talked about different types of media that's available. Lots of different things we can grow in a lab. Lots of different types of media to try to grow those organisms. Some examples of complex media, selective media, differential media is what's coming up. So an example of selective media, the definition, it has some substance in it that either favors or inhibits the growth of something. So if we want to select for a type of organism or we want to select against it, we can grab this one type of selective media. An example showing up here, because we're going to use some selective media in our lab this semester, is a nutrient auger is meant to really just, again, grow anything and everything, like the blood augers that we use. We want to grow anything and everything. Nothing's really inhibited. But a selective media example would be the Sabarot auger that's here on the right. And the inhibitor, the substance that we've put in there that's an inhibitor, is we've added acid to it. We've lowered the pH. That lower pH inhibits bacteria growth, which means we are now selecting for fungus. If we suspect a patient has fungal growth, some type of fungal infection, say on their skin, we know there's bacteria on their skin, and bacteria and fungus will compete for each other, and generally bacteria wins out when it comes to growth on a plate. But if we can inhibit the bacteria from growing, because they cannot grow in that low pH, the fungus can. And so now we can actually see fungus growing. So it's selective because we are selecting for fungal growth by having an inhibitor of bacteria. Another type or grouping of media is differential media. And again, it's called differential because there's going to be some type of change to the media. It's going to look different based on some type of reaction that's happening. And our blood augers are actually considered to be differential media. The red blood cells that are in there, because some organisms, some bacteria can eat up the red blood cells, some can't, and then some can eat them up just a little bit. Because of that component that's in there, and because it now will show us different types of characteristics color-wise, our blood augers are considered a differential media as well. McConkey auger is an example of something that is both selective and differential, which means there is an inhibitor that's added in there. So not everything will grow. Certain organisms are inhibited. And so this is showing an example of E. coli and Staph aureus, both growing, super happy on a nutrient auger because nutrient auger is meant to grow everything and anything it possibly can. But these two bacteria, if I put them on a McConkey auger, which is a selective type of media, not everything will grow. Staph aureus is inhibited. So there's something in here that's inhibiting Staph aureus, which has no effect on the E. coli. But it's also a differential media, is because we can have multiple different organisms just happily growing on it that are not inhibited, like Salmonella and E. coli. Both are growing, but it's also differential because there is a component added to the McConkey media that's going to show us a color difference. And in a McConkey auger specifically, because we'll use these in our lab this semester, what we're looking for color change is, do the bacteria, do the colonies actually look pink? Because E. coli generally don't show up as a pink bacteria unless they're specifically on this media. So we've got that color difference. So media can be both selective and differential. And media doesn't have to only be on plates. It can be in tube media as well, whether it's in some type of kind of augury material in a tube or if it's in a liquid. But we can still add some type of substrate to the media, and whether the bacteria do something with it or not can produce a color change. Transport media is, again, its job is just to get a specimen from the lab or from the patient sample collection to the lab as fast as possible. It's meant to protect that sample. It's meant to just keep the organisms alive. It's not meant to grow it. It's not meant to kill it. It's just meant to keep the organisms alive for a minimal amount of time. And again, the faster you can get samples to the lab, the more accurate the results will be. Now, some special culture techniques. Again, some organisms cannot be used and not be grown on our normal media. And so if we want to grow certain organisms, we can't just pull out a plate. We can't always just pull out a tube. So we've got some other special techniques to try to grow those organisms so we can study them, analyze them, test them, things like that. One is animals and cells. So viruses cannot grow on their own. They have to hijack a host cell. So if we want to grow a virus, we first have to find the cell that that virus can infect. We grow those cells up, and then we can expose the virus to those cells. The viruses will hijack those cells, and those cells now become virus-producing factories. The bacteria that causes syphilis and leprosy, we cannot grow in a lab. It's too artificial of environment. The bacteria are not going to happily grow. However, we can use certain animals to grow them. For example, the armadillo. It's a random animal, but the armadillo is one of the most common animals that can grow both syphilis and leprosy. So you can, if you have contact with an armadillo, get syphilis from it because it is a carrier for that particular bacteria. And it's not uncommon, and we've heard in humans getting syphilis from contact with armadillos. In enrichment culture, it uses a selective substance that enhances one particular, I don't want to say it's a limiting factor, but it enhances something that that particular organism really needs to grow. So if we know an organism maybe needs a very unique sugar to make it happy and to grow it, we can add a lot of that one particular sugar. We can make sure that that bacteria has everything it needs, including that limiting factor, and we can try to grow that particular organism. So some organisms do have one limiting factor, one specific nutrient that they really need to grow happily, and we can ensure that they have that in an enrichment culture. A low oxygen culture, there are some organisms that will grow in oxygen, but they would really prefer a lower amount of oxygen than what's in the air around us. And we can use a candle for that, a candle jar for that. We use it in our lab this semester as well, because some organisms, if we want to make them happy, we want to give them the environment that they grow best at, is we can remove some of the oxygen so that they have that low oxygen environment. They still need some, but less oxygen. And then there are some organisms that need to grow anaerobically, which means if you expose them to the oxygen in the air around them for any extended period of time, they cannot survive. And there are some organisms that, again, that are anaerobic that cause human disease. And so if we want to grow them and identify them, we have to grow them in something called a gas pack jar. So this is showing a gas pack jar, and we have this up in our lab, is that we can put plates, we can put tubes, we can put anything we want in it. But before we seal it up, we open this thing called a gas pack. It releases hydrogen gas, there's your gas, and it will bind up any of the oxygen in this tube. And when you mix hydrogens together and some oxygens together, it does produce water, which sometimes we'll see as condensation, but it binds up all of the oxygen. So the air itself goes into an anaerobic environment. The candle jar, again, we can put plates and tubes in here, light a candle, and close the lid, and eventually that candle will go out. Not because there's no oxygen, but because there's less oxygen, not enough oxygen to sustain the flame. Switching gears a little bit, if we're just looking to not grow organisms, but preserve them, some different ways we can do that. One, we have refrigerators that decrease in temperature, can preserve organisms for a longer period of time. This is why we have refrigerators in our lab, why we have refrigerators in our prep room, is that we can pull our bacteria out of the incubator, put it in the refrigerator. That doesn't kill bacteria, but it slows their growth. So it's almost making them go dormant, but they're not completely dormant. We're just kind of keeping them alive in a low growth state for a short period of time. If we want to keep them for a longer period of time, we can deep freeze them. When deep freezing, this is in Celsius, minus 50 to minus 95 degrees Celsius, the most common temperature is minus 80. So we do have a couple minus 80 freezers. These are specially bought. These are not your freezers at home. Minus 80 freezers that we can preserve samples for years and years. If we want to preserve something even longer than that, say like decades, we can do a process known as lyophilization, which removes the water. Because water, when you freeze it, can expand, which can damage some cells. And so if we want to make sure and ensure that these organisms survive for the longest period of time, we can remove the water as well. It's freeze drying, essentially. Now switching gears again, not just growing organisms or preserving them, but looking specifically at how they grow. So bacteria grow by a process known as binary fission. Again, fission means to split. It's the opposite of fusion. Binary meaning two. So the cells will start to double everything in them, and then they will, as they grow and as the organelles, and there's not a lot of them, but as all their essential organelles double, they just divide into two. So you have one bacteria, then you have two bacteria, and then these two bacteria will each grow, divide, and then you have four bacteria. So it's a constant doubling. So this again is showing, here's one bacteria, it's growing, it's doubling, it's divided. Now you have the two bacteria. And then those two bacteria will grow and divide, and now you have four, and four become 8, 8 becomes 16, and so on. The time period that it takes for a bacteria to grow and divide is called the generation time. Sometimes also called the doubling time, because that's the time it will take for then it to double, because it's divided into two. Depending on the bacteria species, and depending on the environment that that bacteria is in, does it have all of its nutrients? Does it have a nice warm temperature? If a bacteria is in its optimal growing environment, it's not uncommon to have a generation time of 30 minutes or less. That means the bacteria within 30 minutes and less can completely double. Another 30 minutes will double, another 30 minutes will double, and you can get a very, very fast increase in number of bacteria. It's called a logarithmic growth when you have this constant doubling of bacteria. It looks like it's growing kind of slow at the beginning because it's 1 becomes 2, 2 becomes 4. But then as you start to get those larger numbers, and those larger numbers just keep doubling, we get a very, very quick growth increase of number in bacteria in a very short amount of time. Now, my little math problem on here. So for example, if bacteria have a generation time of 5 minutes, that's pretty short, and you start with 10 bacteria, because you have to have some bacteria to start with, how many bacteria would you have after 15 minutes? So doing a little bit of math, and I'm one of those, it's best if I write it out, so I wrote it out. So if we start with 10 bacteria, and again, our generation time is 5 minutes, and we've got 15 minutes. So after one generation time, after 5 minutes, 10 became 20. After another generation of 5 minutes, those 20 become 40. And after another generation time of 5 minutes, so 5 minutes, 5 minutes, 5 minutes to give us our 15 minutes, those 40 become 80. And if we want to say what if it was 20 minutes? Well, those 80 would become 160. And if it's 25 minutes, those 160 would become 320. So we start small, but then we start to get big numbers real fast. And that's every 5 minutes. As bacteria are growing and doing this binary fission, there is a growth curve that does happen. When we put bacteria on a plate, they don't right away that second start growing. There is this lag phase that occurs, and they're adjusting to the environment. They're breaking down the nutrients that you gave them. They're just starting to do something. They're just kind of adjusting to their new environment. Then, once they've adjusted to their new environment, they made enzymes that broke down the nutrients that you gave them. That's when we start to see that logarithmic growth phase. They are rapidly growing and dividing, growing and dividing. It is during this phase that we pull them out of the incubator, because they are all happily growing and dividing, and happy bacteria will gram stain more accurate. They're going to be alive. They're going to gram stain better. It's the best time to pull them out of the incubator. If we let them sit in the incubator for an even longer period of time, because it's usually about 24 hours, is while they're in this logarithmic phase. Then they start to go into a stationary phase. There's only so many nutrients for so many bacteria that are on that plate. And as bacteria are breaking down nutrients, they are producing waste products. So now we're starting to get into an environment that's not the greatest environment. So some of these bacteria cells will start to die. Doesn't mean new bacteria aren't being made. There are still binary fission occurring. However, now we're also going to have dying cells. So it's stationary because number-wise, if you're getting 100 new bacteria during this time period, there's also then 100 new bacteria that just died. So overall numbers are not increasing. And dead bacteria do not always gram stain accurately. So we try to pull them out of the incubator before they get to this phase. Then they do eventually, if we left them in the incubator even longer, go through the death phase, which just means more bacteria are dying than growing and dividing. There are still some alive, but they're just dying faster than new ones are being formed. Now that we've grown them, we've made them happy, the next question is, well, how do we measure how many bacteria are present? How do we measure that we have growth? How do we measure how many bacteria are present? So we divide our different measuring of microbial reproduction into two different groups, direct methods and indirect. Direct methods that require incubation and ones that don't require incubation. Again, incubation is more time consuming. So a direct method that does not require incubation. We've got a couple different electronic counters that can count bacteria numbers and give us a pretty good approximation of how many bacteria are in a solution. The first one is called the Coulter counter, is that we can put bacteria in a broth, in a fluid, and the more bacteria cells there are, we put this electronic device in the fluid, but there is a gap, and anytime a bacteria cell interrupts that electrical flow, that electrical current, the instrument records that as a bacteria cell. So the more bacteria cells that are present, the more interruption of that electrical flow. And it can actually make a pretty good guesstimate of how many bacteria are actually present, say per milliliter that's in that fluid. Flow cytometry is very similar, except instead of interrupting an electrical current, it interrupts a laser light source. So we can still put bacteria in a fluid, we can shine a light through that fluid, and anytime that light hits a bacteria, it will bounce in a different direction, and there's an instrument that's recording that. So the more bacteria cells there are, the more bouncing of that light source, and you're getting a little more estimate of how many bacteria cells are actually in the fluid. Another direct method that does not require incubation is counting the bacteria cells underneath the microscope. So there are specialized slides that are meant to do this. Etched on these slides are grids, and you can put a drop of fluid on this etched grid on this slide, put a cover slip on it, so another glass covering on there, and then we know exactly how much fluid is between the cover slip and the slide itself, and underneath the microscope we can actually count how many bacteria cells are present. And because we know the volume of how much fluid is in between that cover slip and the glass slide, we can also get a pretty good estimate of how many bacteria there are per milliliter. It's putsy, but this is actually quite cheap. You can buy these slides, they're reusable slides because you just rinse them off between each one, and it's a pretty cheap way to do it. It's time consuming because you have to sit there looking through the microscope counting cells, but it gives you a pretty accurate estimate without needing any kind of expensive devices. Some direct methods that do require incubation for giving us bacteria counts these are my next three slides too: serial dilution and viable plate counts, membrane filtration, and a most probable number. So serial dilution and viable plate count. If I've got a broth of fluid, we're going to start with my original, and I can see that it's cloudy, it's turbid. If I tried to put a sample of that on a plate, there'd be so many bacteria, it's too numerous to even count, even make a good guesstimate at how many bacteria. but we can start diluting that sample. So this is showing where we took 1 milliliter of our original culture, put it in 9 milliliters of a sterile broth. We incubated, or we put a sample of this on a plate and incubated it. But instead of just then waiting for that to come back, we can do other dilutions as well. So this is a 1 to 10 dilution because I took 1 ml of my original sample, mixed it with 9 milliliters of broth. I have a 1 to 10 dilution, and that's still too numerous to count. But I can further dilute this. And you don't know at what point it's too dilute that you won't get anything to grow. So it's not uncommon to do a 1 to 10 dilution, a 1 to 100, 1 to 1,000, even a 1 to 10,000. If it looks really cloudy, they can also go out to that 1 to 100,000. But noting, this is time consuming for all these dilutions and incubation. And not only is it time consuming, this is a lot of resources. You've got to have a lot of tubes that have sterile broth in it, and you have to have lots of plates. And then after incubation, you still have to count. But we can still go back and do the math. If I have a 1 to 10,000 dilution, and then I still only put 0.1 milliliter, so I don't even put a full milliliter, so this is actually another tenth of a dilution because I'm taking a tenth of a milliliter, and I put that on a plate, I can go back and do some math and say, well, there's 6 colonies times 10,000 times another 10. So ultimately 6 times 100,000. I can say there's 600,000 bacteria per milliliter. And if I go back one and I do the math and say 65 times 1,000 times another 10, so 65 times 10,000, well, it's 650,000 bacteria. We're getting a pretty good estimate of how many bacteria there are. This just isn't done all that often just because of the time and the supplies. So it costs money, it costs time, but you can still get another pretty good accurate estimate of how many bacteria there are in a sample. Membrane filtration is usually used if we know we've got a small number of bacteria in a very large volume of fluid. So this is commonly done, say if they wanted to take an environmental sample. I want to know how much bacteria, say, is in a lake. Well, I'm not going to take just one little tiny milliliter sample. I may take a gallon or a liter of that fluid. And because I want to know how much bacteria is on that liter, I can't take the whole liter of fluid and pour it on a plate because that's just not going to fit on there. But I can take that liter of fluid, run it through a vacuum, and then any bacteria are going to get stuck to a little filter. It's almost like a little filter paper grid. The bacteria are going to get stuck to the filter. I can take that filter, which has bacteria now stuck on it, put it on a plate. And any bacteria that are stuck on that filter paper are going to grow because we give them all the nutrients they want. And then I can go back and I can start to count colonies. Oops. So I can say, okay, well, I counted 80 colonies and I put 2 liters of fluid through here. Well, that means there's 40 colonies or 40 bacteria per liter. So if you've got a large volume of fluid and I want to know how many bacteria are in it, we usually filter them out. Now, if a microbe can't go in any type of agar and it has to stay in a liquid, which means we can't just look, at bacteria cells and count them on a plate. There is a process known as the most probable number. Is we can take an undiluted sample, we can start to dilute it out. We can then incubate the tubes, but we put them in a very specific type of media. And the more bacteria there are, the more of the tubes, because usually we put them in 5 tubes each for each dilution, the more bacteria there are, the more tubes will turn colors. And we know based on our dilutions, based on the number of tubes that turn colors, we've got mathematical tables, because the math is pretty complex. We've got mathematical tables that will just say, okay, well, based on your dilution series and based on how many tubes turned positive based on the dilution series, we've got tables that will tell us an estimate, a number of bacteria that are there. Now, some indirect methods that indicate the growth has occurred. That doesn't give us a straight up number, but we can see that bacteria have occurred. The first is metabolic activity. So the more bacteria cells there are, the faster a substance like a sugar or a protein can get broken down. So we can look at metabolic activity. The more bacteria, the faster something occurs. We can look at dry weight. We can filter these bacteria out. We can dry them and we can weigh them. Again, they don't weigh a lot. They're microscopic, but if you get enough of them, we can actually weigh them out. They do still have mass, and we can get an estimate, a number of bacteria. We can look at genetic methods. We can look at DNA. The more DNA that is present, the more bacteria there are. And we can also look at turbidity, which is generally what we do in our lab. The more bacteria there are, the more cloudy that sample is. There are some machines that can measure turbidity and cloudiness and give it a number. This is just looking at something that's more crystal clear versus something that's more turbid or cloudy. And we've got machines that will shine a light through it. If it's super cloudy or turbid, the light doesn't shine through directly through it to the sensor. Instead, it's bouncing off in other directions. And so this gives us, how turbid it is based on how much light can actually shine through it. Again, the more turbid something is, the more bacteria it is. It's not going to give us a number of bacteria, but we can start to compare turbidity. We can have two tubes that may look like they have about the same turbidity, about the same bacteria numbers. We can put them in these little machines and it will tell us which one's more turbid, which one has more bacteria. Because what our eyes see is not always an accurate estimate of how many bacteria are in there. So we're going to end right there. We've got our different methods of measuring bacteria, different methods just looking at binary fission and just kind of how those bacteria growth. And then I'll have another PowerPoint for some of that introduction on chapter 8 material.